Welcome to the first seminar in our virtual seminar series for the School of Chemistry and Biochemistry. So it's really great to have all of you here with us. I think it's going to be a lot of fun to talk science for the rest of the semester virtually, and we're lining up a great set of speakers for the rest of the semester. There's still a sign-up sheet going around, so if you'd like to present, we'd love to have you do it. So please sign up to present if you're interested in. Our first seminar as part of this virtual series is going to be focused on what our department is doing to help respond to the COVID outbreak. And so anecdotally, I've heard, various labs are involved in these efforts. So I thought it would be nice to have Professors Williams and Finn talk more holistically about what exactly people are doing and really highlight all the important work that's going on. And so it's really great to hear them talk, and I want to thank them for taking the time to tell us what the department is doing. If there are questions, please post them to the chat, and then we'll read off some of the questions after the speakers are done. So with that, Professor Williams, please start when you're ready.
Okay. I'm sharing my screen. I can. Okay, hi, I'm Loren. I'm presuming my screen is being shared. Maybe Amit, can you confirm that? Can somebody confirm that to me?
See your screen, yeah.
Okay, great. Okay, so as some of you may know, we generally think about the ribosome and nothing else. And about 3 weeks ago, we sort of got distracted. And I'm going to explain to you guys, to the best of my ability, sort of what we got distracted by and what we are doing. Just to start with, I'll say that seems all kind of blurry now in the past, but I don't know, maybe 3 weeks ago or something, I just decided that we could do something, and so that we should do something. And I didn't have such a clear idea of what to do, but I just decided I had to do something, and so this is kind of where that led. So what we are part of this organization called the State of Georgia COVID Testing Task Force, and this is an organization that comes out of the governor's office and actually is being run really through the National Guard. So I'm dealing with military guys. We get along great. We talk every day. We have a 1 o'clock sort of conference call where we go over things, and people involved in this are the testing labs. They're called CLIA labs. There's a lab at Medical College of Georgia. There's one at Georgia State. There's some at Emory. There's various labs, and this is part of an effort to get these labs up and running efficiently at basically the highest throughput possible. And this is very complicated to do this. I mean, the lab at MCG was already up and running and doing testing already. The lab at Georgia State was really just an academic lab. They had the legal authority to do testing, but they were really just an academic lab studying viruses. So that's, building that into a testing lab, to do high throughput testing quickly and efficiently is the analogy that comes to me. It's kind of like waiting till your house is on fire and then saying, I think we need a fire department. That's kind of what's happening. So we're sort of trying to build a fire department while the house is on fire. So specifically what we're doing, and I'm going to tell you who we are in a while, but what we're doing is we are ensuring the supply line. We are ensuring that there are reagents. This is what we're supposed to be doing. We're supposed to ensure that there are reagents, that there are enzymes, that there are primers, that there are probes for the testing that is going on at these CLIA labs. And of course, you may have heard this, there is an acute shortage of everything, of plasticware, of enzymes, of everything. There's just shortages of everywhere nationwide. So we are using Georgia Tech facilities and personnel, and we're producing. And starting, I think Monday, we will start to actually distribute our first kits to Georgia State. This is what we're supposed to be doing, but really what we're really doing is a little bit different. We are preparing these reagents, and we're doing them, we have many parallel pathways going. We are taking what's called the TAC-PATH kit. That is a very highly optimized, very sophisticated kit produced by Thermo Fisher. I'll explain to you a little bit about what it is. And we are re-engineering that. We are figuring out all the enzymes. It's a very complicated kit, and we are re-engineering that so that we can supply an equivalent kit to the testing labs. But on top of that, we're working with people who have already done a lot of background work to make new kinds of kits. For example, Andy Ellington at UT Austin has made some fusion proteins which have very nice properties, a reverse transcriptase fused to a thermal stable polymerase. And we're using his enzyme to try to establish an alternate kit. There's something called the CDC probes and primers, and we are engineering those, converting them from singleplex to multiplex. We are working with the World Health Organization probes and primers, and then there's also dye-based assays. Basically anything to do with RT-PCR, we're trying to figure out how to make it better, cheaper, faster, easier, and more available. But on top of that, we're dealing with the National Guard, and there's some very smart, very dedicated people who we're dealing with, but they're not sort of so technical. And so we have taken it upon ourselves to be their technical advisors. And we have done everything we can to learn about sample collection, sample transport, sample prep, the RT-PCR, and the FDA regulations. And so we advise them on this. Sometimes they don't like it, but we do it anyway. We try to provide them with the best technical advice we can give to them. So, all right, let me tell you who we are. There's a lot of people involved in this. It grew very quickly from like me to, I don't know, maybe 30, 40 people. I actually don't really know. We have divided our structure up. You can see on the left-hand column, this is sort of how we are broken up, and we're totally committed to Microsoft Teams. And everything we do is through Microsoft Teams. So we have a communications group that's led by Jennifer Glass. We have, of course, Sue, who takes care of finance, but she kind of helps us with a lot of the regulatory and all the paperwork. There's somebody on here who a lot of you may not know, who is Sam Machu, and she is actually, that's Julia Kubanik's postdoc, who just kind of came early on and said, can I help? And turned out to be like this outrageously hyper competent person who can do a lot of things. And she's in charge of formulation. That means she's really the person doing the final testing of our reagents, etc., before we assemble them. And Nick Hudd is in charge of our probes and primers group. That's, of course, all nucleic acid based, and Nick is one of the world's experts in nucleic acids. Vinny is in charge of reverse transcriptase, and I think Vinny has purified to homogeneity like $1 million of MMLV reverse transcriptase. We have, of course, Pamela is helping us with safety. We have all kinds of, we brought so many different kinds of nucleic acids into Georgia Tech so fast that the protocols and everything have to be, there's an enormous amount of paperwork and regulatory things we're dealing with. And Sue is helping with that. And also, you can see sitting next to Pam is Jessica. Jessica is my lab. She's a research scientist who works for me, and she's helping us with many aspects of this. But she is helping with the safety. And then in the bottom right of the slide, you can see Raquel and Bettina. And these are like, I mean, if you are trying to purify protein, you want these people. I mean, they are like a force of nature that is just kind of incredible. So they are they're in charge of our TAC group, which is one of the enzymes that is not so easy to purify, and they're working on that. And then you can, I guess it's kind of cut off on my screen. I don't know if you guys can see it, but at the top right of this, you can see Julia Kubanik, MG Finn, and Andres Garcia. They're cut off on my screen, and I appointed them. I didn't even ask them. I don't even know if they know this, but they are my advisory group. And so if something bad happens and I want to complain, I send them email, and they either pat me on the head or, I don't know, kind of, if they don't do that, you might go to jail. So this is sort of the structure of, this is part of the structure. You can see we have a regulatory, we have some people who are doing nothing but reading FDA regulations, trying to figure things out. We have people working on all aspects of this problem from the ground up. So this is a lot of the students and postdocs who are working on this. Not all of them, I've sort of stuffed them all together here. I'm not going to go over all their names. A lot of you know these people, but this is just an amazing team. You cannot believe how fast these people are working, how hard they're working, and the kinds of problems. It's like no problems is unsolvable. It seems like whatever problem we have, and we've had a lot of problems, somebody says, oh, I know somebody who can do this, and then a new person joins our group. So that's sort of who we are. And then we work through blue jeans. We have blue jeans meetings every day pretty much. And we have learned to love and hate blue jeans, but it works for us pretty well. So we have developed some sort of new ways of interacting with people, because what we have is we have new ways of working. Number one, we don't want people close together. We don't really even want anyone in the same room as anyone else. There's the whole social distancing thing that we're supposed to do, but there's also the issue, if somebody comes down with COVID, then anybody who has been in the room with that person has to go into quarantine, and that will shut us down. So we don't want two people in the same room pretty much ever in this project, which is pretty difficult. And so one of the things we do, we have a check-in and check-out system so that we all know who's where. And this is just a page from one day. You can see various people coming in and out of the lab. And we make sure we have sort of an electronic buddy system. This has been worked out with Pamela, so that people can kind of be alone in the lab, but not be alone, because of course we don't want people working alone, but they have to work alone. So we're kind of inventing some new ways of doing things. We're working very hard to be safe. We don't want any student to get sick. We don't want anybody to get sick. So we're trying to think very hard about how to ensure safety. Okay. So now I want to talk about a little bit about the technique we're talking about. And this technique is based on Watson and Crick base pairing. It's really, everything I'm talking about here is Watson and Crick base pairing, meaning G pairs with C and A pairs with T, or U. They can pair with U too. So that base pairing is kind of built into everything we do. And of course, you guys remember your biochemistry, those of you who've had my class, or Nick's class, this is a very, very specific interaction. And you can use this to find things. You can use this to detect things. And that's what we do. So the system we're focused on here is called RT-PCR. And this is an incredible technique. I think from analytical biochemistry, it's almost, I'd say, reached a level of perfection in the sense that the amplification is just incredible, of course. The selectivity is, of course, incredible. And and then it's linear at the same time. That's what I'm going to show you. So this system was invented. Actually, this was invented during the HIV crisis, when number one, it was important to find very low titer virus in people, and number two, the actual titer of the virus, like how much virus is there, was very important. It turns out for COVID-19, we don't really care how much virus is there. There's generally a lot, and it's a yes or no question. But for HIV, it was very important to know how much virus, and to detect very small. That was a virus which was very good at hiding. So really, RT-qPCR is a bit overkill for what it's being used for here, but it is a totally marvelous technique. So the RT stands for real time. I think it should stand for real time, but it also sometimes stands for reverse transcriptase, which is part of it too. So I don't actually know what the RT stands for. The Q is quantitative, and the PCR is polymerase chain reaction. So I'm just going to go through this really quickly and just explain some of the, I think, really salient features that I think are really, really interesting. Okay, first thing is, the first part of this is an RNA virus. So we're detecting RNA, and you can't PCR RNA. That doesn't work. So the first step of this is a reverse transcriptase that sits down on the RNA and converts it to DNA. And this is the first level of selectivity, because see that green, that's a primer. And we put the primer in there. That primer is specific for the viral RNA. So there's a lot of RNA in these samples, right? There's all the host RNA. There's a lot of RNA from the human that it comes from. There's all kinds of RNA, and that primer is specific. So it sits down and it makes DNA. That yellow is the DNA copy. And this is a linear amplification. And what I mean is that enzyme sits down and it makes a copy, and then another primer can get in there and it can make another copy. So you can make enormous numbers of copies of cDNA from one RNA. So this is not an exponential amplification, but this is the first step in selectivity, and the first step in amplification is here. And this tends to happen at moderate temperatures. And once you warm it up, so you let this reaction go for a while, and once you warm it up, the reverse transcriptase is dead. So you can kill that reverse transcriptase when you want to. And generally, we let this reaction go 10 minutes, something like that. Then you warm it up and kill it. Then you have the DNA, and the next step is the DNA amplification. And this, I'm sure many of you are familiar with this. But you start with the DNA double strand, and this shows a short fragment, but really the DNA, a lot of times we're looking at, is very long. And you select for certain sizes, again, by primers. So these are different primers than you use for the reverse transcriptase. There's a set of primers. And so initially nothing happens. Then you heat it up, you melt the DNA. Then you cool it quickly a little bit, and the primers bind. And as soon as those primers bind, the thermal stable polymerase sits down and starts to extend. And this is in the elongation step. They're not showing it completing, so it's not all the way to the end. But if they showed that, you would have blunt end pieces of DNA whose ends are defined by the primers. So there's two things that are happening. Number one, you are searching, because the primers search and only bind to the DNA that you specify. And number two, you are amplifying. So every time you go through this cycle, and it ends up being the cycle of 1, 2, 3 here, every time you go through that cycle, you double the amount of DNA. So if you start with 100 strands of DNA and you do this 30 times, you have 100 to the 30. It's just an enormous amplification that you get out of this. Okay, then the next step, and this is I find totally fantastic that this works. You have what's called a probe, and a probe has a fluorophore and a quencher. So normally it's dark. You can't see it because the quencher is close to the fluorophore. But once you've amplified the DNA, then this probe sits down on the DNA. And this is your third level of selectivity, right? This probe only sits down. Again, it uses Watson and Crick-based pairing So the probe only sits down on the DNA that you have specified, which is the DNA in your amplicon that you're amplifying by PCR. So this is the third level of selectivity. And what happens is the polymerase has this, what's called the 5' to 3' exonuclease, and it comes in and it sees that probe and it cuts it in half. So this turns on the light. Okay, so once the polymerase sees that probe on the DNA and cuts it, then the quencher is far away from the fluorophore and it turns it on. So think about it. You're starting with a little bit of RNA. You make that into DNA specifically, right? Because you've used a primer. So you're only amplifying, or you're only making the DNA you want out of the RNA. Then you're amplifying just select DNA out of that, and that is also specified by those primers. And then here's the third level of selectivity, which is your probe. So at every one of those, you are selecting for sequence. So it is very difficult, if this thing is done right, for you to get a false positive. Every one of these is a way of screening that out. Okay, so I want to back up for a minute here, because what we have is a pair of primers, and they're green. But imagine you had a different set of primers here too. You could do what's called multiplexing, where you run different PCRs at the same time, and they kind of don't see each other. So you could be amplifying one fragment of the DNA with one set of primers and another fragment of the DNA with another set of primers. So you could end up not with one base paired fragment here that's been amplified, but two. And you can also have multiple probes with different colored fluorophores on. So this is called multiplexing. And so you can look at the same time for different things in your RT-qPCR reaction. This is really sophisticated technology that I think was developed and has been perfected over the years. It is it is so incredibly sensitive and so incredibly specific. And this is the machine you do it in. And we have this, and we have some of the instruments we've have at Georgia Tech have been distributed down to Georgia State and places where we have, doing everything we can to help them get going. But what you see here is a drawer that's open, and this is done in a high throughput format. You have a bunch of wells, and there's optics built into this thing. So where you are doing the thermal cycling and you are doing the optics at the same time, and you are watching to see in real time what gets amplified. So I think the next one is I have some data. Yeah, this is some of Sam's data. And so what you have in this graph is you have cycle number. So in the beginning, you're cycling and something is happening, but you can't see it. It's happening at a very low level, and but it's exponential, and pretty soon the signal starts to come up. And so that's the optical signal is on the vertical axis. And so what Sam has done here is she has done sort of a serial dilution of different amounts of substrate. And this shows you how linear this is. This is why it's called quantitative. So she has these different colors. She did them in duplicates. Does these different color lines. Those amount to different amount of material that she is selecting for. So this is this shows you how sort of linear that the response is from this thing. So so really what we're doing, I'll just go back to this, is we are developing enzymes for this. We are converting the CDC kit into a multiplex kit. That is the thing we're doing. We're trying to have these reagents down there by Monday, where we have re-engineered the CDC kit, which is called the singleplex kit. That means it needs 3 wells in a plate. And if we can multiplex it, then it means 1 well in a plate. And then the whole system becomes much more efficient. So they're dying for us to convert the CDC singleplex kit into a CDC multiplex kit. That is our first task. Which, as soon as this meeting over, I believe Sam will, I can call Sam and say, is it a go or a no-go? I think she's doing those experiments as we speak. We have made everything and we're just testing it. So yeah, that's what we're doing. And these are the people who are doing it. And I think with that, I will maybe turn it over to MG and let him tell you guys what he's doing. I'm going to stop sharing. Or if I can answer questions,
I think there's a couple of questions, Loren. Professor Matt Torres asked, are there mutations in the primer binding sites? Is there any information known about that from the CDC or anywhere else?
Yeah, that's a really good question. In fact, the choice of primer binding sites is very, very important because number one, you don't want to. In fact, one of the problems with the CDC kit is that one of those primer binding sites actually makes a hairpin that competes. So there's something called the N2 amplicon, and that actually we can see that. Sam can see that, and other people can see that. It gets amplified a little bit after the other one because the primer was not correctly designed. It is competing with a hairpin in the template, and then of course people they have tried to work and say select sites that are not highly variant, and that are not going to change over time. And I think at this point that has not been a problem with any of the primers. But I think ultimately, that of course, as this virus mutates over time, there will be probably issues with some of the primers. We did look. Anton Petrov is sort of in charge of bioinformatics, and he does have a map of kind of rates of mutation over various parts of this virus. And I think we looked at that, and I think the primers in general look pretty good.
I've got a question. So I remember the week of March 16th. So that was like when we went from things will be fine to, oh shoot, everything's going to shut down. And so I'm curious, were you in these higher level talks with the state prior to that week, or did everything come down that week or the week after?
It's hard for me to remember. All I can remember is that this is what happened, is I decided that we had to do something. And, the thing, like Santi in my lab has done so much RT-PCR, we do have expertise in this, not in diagnostics, but we know some things about RT-PCR. So I said, okay, there's going to be a problem. I just said, there's going to be a problem. This is not going to work because things are not set up. So we have to start doing something. And that was like before anything. And so then we had some meetings. And then really, honestly, what happened was they said, okay, so there's going to be some structure. So put everything on ice and let's wait until we are told what to do. And I just said no. And I wrote an email to the vice president of research, and I said, I think I was just kind of upset. I said, I don't care. I'm doing it. We're going to push ahead. And, so I did that. And then that was really weird. On a Sunday, then I got a call from the president of Georgia Tech who said, I heard there's some things we can do. The governor has called and asked us if we can do something. And I heard there's something, and I said, yeah, we can do it. And so then he said, okay, go for it. So that was sort of how it happened. There's a lot of things. Julia Kubanik and the dean's office, when I said I'm doing this anyway, they were like, who's going to pay? And Julia said, okay, we'll back you on this. So somehow enough people kind of came together that we just kept on pushing. I wish we'd started sooner. We'd be able to help sooner. We'd be in a better place, but it could have been worse, I guess.
Professor Kubanik asked, it was on March 12th that Loren said we have to do something. On March 14th, the state asked for GT's help. On March 19th, Loren was asked to join the task force. So okay. Thank you for clearing that up. There's a question from Ashley Chapman that's asking, can you talk about the risk of false negatives in this testing scheme? Where can the test fail?
Actually, that's a really good question. The risk is not in false negatives, it's in false positives. This test is like, this is like heating a baby bottle with a blowtorch, I think. This test was designed and optimized with HIV and can detect, at its limit, it can detect 20 molecules of viral RNA. And really, these the samples they're testing, there's billions and trillions of viral particles that end up being sensed. So the primary source of error actually is in swabbing and taking collection. The PCR is generally it's really an exact thing, and it really doesn't tend to make mistakes. It's really overkill for this. I mean, it's good to have overkill, but it is. In fact, yeah. One of the things that I'm convinced of is that we could make a simpler test that would be cheaper, easier, faster, and would not really give substantially higher error rates, because this test was designed for something that is much more difficult than what it's being used for.
Thank you. I think with that, thank you, Dr. Williams, Professor Williams, for the talk, and we'll switch over to Professor Finn.
Okay.
So through a miracle of modern technology, I was unable to project slides from my computer, so Sue is going to do them, and we're going to do this seminar the old-fashioned way. Most of you are too young to remember the days when the commanding lecturer would ring out in authoritarian tones to please change slides. Next slide, please. So we will make an effort at doing that without the authoritarian impulses. Thank you, Sue. Okay, I'm going to tell you about really a tremendous amount of work being done by graduate students Ashley Chapman and postdoc Langjun Zhao and colleagues at the CDC. And I'll tell you how this project got started. I won't tell you about an effort that's going on with Emmeline Blanchard in Phil Santangelo's lab and Justin Lawrence from Brittany Schmidt's lab and others on testing disinfection methods. That's something that Phil and I started several weeks ago and is really, I think, proving to be somewhat useful. But there is a whole, for those who are interested, there is a large number of people associated with Georgia Tech that are working on disinfection methods, sourcing them, figuring out how to scale them, figuring out how to test them, because that too is a need in Georgia, in rural Georgia particularly, but elsewhere in hospitals all over the world. So a lot of that is going on as well. But today I will tell you about our efforts to develop a anti-coronavirus immune response. They're certainly not alone, but we have the aid of really some tremendous people at the CDC. So next slide, please. So, and I should mention right at the top, I stole most of these slides from Ashley, who prepared them for a group meeting last week. So Ashley, thank you very much. And really, she should be giving this talk because she knows far more about everything than I do. But I get, as is common with PIs worldwide, I get to steal the thunder of those who actually do the work. So my apologies to Ashley and my tremendous gratitude for the work that she and others are doing. So here's April 1. This is the website that Johns Hopkins has for tracking COVID-19 cases. You can see almost 1 million confirmed, and those numbers there. And then go ahead and go to the next slide. This is 2 weeks later, April 16th, more than 2 million cases, 138,000 deaths, 3 million tests in the US. No, I'm not sure what that right column is, sorry. But the point here is that, as we all know, things are developing rapidly. COVID-19 is the disease name. Of course, the causative agent is SARS-CoV-2, severe acute respiratory syndrome, coronavirus 2, to distinguish it from the first SARS virus, which appeared around 2003, and the next coronavirus to appear which is the Middle Eastern Respiratory Syndrome virus, MERS, in about 2008. All of these coronaviruses are, of course, respiratory viruses. They infect primarily the lung epithelial tissue, and they cause cytokine storms, huge immune responses, and inflammatory responses. And that is, of course, part of the problem, which then tips over into symptoms that look like viral pneumonia. But this particular virus, for a reason we'll get into in a minute, is quite pernicious. So next slide, please. This is one of my favorite papers. Of course, we are exposed to viruses on a tremendous scale every minute of every day. This paper was published a couple years ago and basically seeks to quantify the amount of microorganisms that are raining down upon each and every one of us every day. And the estimates are that each square meter of the Earth gets hit by about 1 trillion viruses circulating in the atmosphere and about 10 million bacteria just floating around in the air every day. And so the coronavirus is transmitting because it has a particularly efficient way of infecting us. But our immune systems, of course, see lots and lots of viruses and bacteria all the time. So there is some hope that immunity will come to the rescue. We just need to give it some help. Next slide, please. So SARS-CoV-2 coronavirus, you see that iconic image, which I think was first done by the CDC, at the bottom right. And the most striking thing about that image, of course, are the red, beautiful little blobs that come out. Those are known as the ectodomain, or the spike protein. There's actually, each of those little red things is a trimer, a symmetric collection of three of those proteins divided into two subunits, S1 and S2. And the primary interaction that is responsible for the virus being able to infect human cells is the one that's zoomed in on the left. It's an interaction with angiotensin converting enzyme 2, ACE2, which is on the surface of many of our cells, but particularly on lung tissue. And this particular coronavirus, all coronaviruses interact with this enzyme or another surface enzyme. And this particular virus has some mutations from the previous versions that make this interaction with ACE2 particularly strong. And it is thought that is one of the ways in which, one of the reasons for which this virus is a little bit more infectious than the other coronaviruses. Next slide, please. So the take-home message is that spike protein is basically what you see when you're coming from the outside in and zooming in on the virus. And in essence, that's what the immune system sees first. So COVID pathology, I won't spend a lot of time on this because we all know this too well. The incubation time from 2 to 14 days approximately. Of course, there will be outliers. There is certainly a large difference in the degree of severity of infection in terms of symptoms caused. We all know, as our governor learned only recently, that many people can be infected and be shedding virus and not be symptomatic. And of course, symptoms range all the way to the very severe. And the fatality rate of 1.38%, I don't know if that's the newest number. It's probably a pretty good estimate, but I think that number changes a little bit as the statistics get better. So let's go on to the next slide. And Loren notes there's a lag in slide advance. If somebody would just tell me if I'm getting too far ahead of my keys here, I'd appreciate it. In case you don't know, of course, the coronavirus is able to survive, quote unquote. That means remain infectious outside of the body. It is relatively easy to inactivate. Soap and water does it, as most enveloped viruses are susceptible to soap and water. The term enveloped means that the virus shell is coated with a layer of lipid, of cell surface lipids that are taken from the host cells. And so if you disrupt that lipid layer, which is what soap will do, soap and water will do, then you will inactivate the virus. But in the absence of soap and water, the estimates range from a few hours to many hours for the virus to remain infectious on various surfaces. So this is why, of course, we want to make sure that we don't touch hands and that we clean and disinfect surfaces that come in contact with lots of people. Next slide, please. I'll skip this one. We all know, again, the viral infection rate. We don't know what the viral infection rate is, but because testing, as Loren pointed out, has been so inadequate, but certainly it's spreading rapidly all over the place. Next slide, please. This coronavirus almost certainly arose, as other coronaviruses have, from interspecies transmission. Looking at the sequences, these coronaviruses are quite close to viruses isolated from bats, and it's pretty much beyond dispute that the virus arose in China, was aided in human transmission by human-animal contact and lots of high density of both. This virus was not created in the laboratory, and of course has rapidly spread all over the world, so that it's coming at us from everywhere. But the sequences can be used to trace the origin, the species origin, pretty nicely. The note here at the bottom is important. This is what I was alluding to earlier. The receptor binding motif, RBM, is the part of the spike protein that binds to this ACE2 receptor. And COVID-2, the novel coronavirus, has 5 mutations in, or residues, 5 residues that interact with that receptor that are mutated from previous versions that make that affinity, that binding strength to the receptor quite a bit higher. And that is, again, thought to contribute to the infectivity of this pathogen. Next slide, please. So how can you fight the virus? And of course, one of the wonderful things about this is, like Loren, lots and lots of people are saddling up all over the world to bring their expertise to bear. A virus like this is relatively simple, but it has a lot of things that go along that make its replicative capacity happen. The cartoon here is a nice one. It shows you the major components of the virus in terms of their function. You see the trimeric spike sticking out from the surface. There is another membrane protein that helps hold the membrane to the particle, the protein shell, an envelope protein as well. There's the RNA genome. So this is a positive sense RNA genome. We'll explain that in a minute. The RNA is what codes, what has the instructions for every component of the virus. That is bound to protein that is evolved to essentially complex to bind to that RNA and carry it around. These are the structural components of the virus. The virus also has genetic instructions as part of the RNA genome to make 16 other proteins that contribute to the function. And the major drug targets derive from those proteins. So hit the button again, Sue, and you'll bring up display on the left, which shows the virus life cycle. And it's maybe a little too small to see. I apologize for that. But if you start at the top left, the virus binds to the ACE2 receptor, as we talked about, and then some magic occurs, and it fuses with the cell membrane, and its RNA protein complex, the nucleoprotein, gets released into the cytoplasm of the cell. Exactly how that happens is actually not known, but the ACE2 binding is important, is essential. If you block ACE2 binding, you block infectivity of the virus. And so, we have one of many potential ways to fight the virus. Let's block ACE2 binding, for example. The fusion event can also, this magic occurs, that can also potentially be inhibited. Then the RNA gets into the cell, it gets translated into protein, and then that protein is two large polypeptides, which are known as the polyprotein. They get cleaved by a protease enzyme that the virus also encodes. So there's another function that can be attacked. You stop that protease cleavage, you stop the maturation, the creation of the infectious virus. Then a lot of proteins get together to make a transcriptase complex, which goes ahead and generates a whole bunch more RNA. And again, there are functions there, such as primases, there's RNA binding. I'm reading here these blue at the lower left. There's an RNA polymerase, which chunks out much more RNA, an RNA-dependent RNA polymerase. There's a helicase, exo and endonucleases, methyltransferases. All of these enzymes are coded by the virus. They're not in the host cell, and they are required to make huge quantities of viral RNA and to chop that RNA up into the messages that code for the various proteins that are required to make the structural proteins, which the cellular ribosomal machinery does. And so the virus, of course, like all viruses, is very clever in hijacking cellular machinery and bringing along what it needs if the cell doesn't have it to make more of itself. So each of these functions is a potential drug target. And there are many, many people around the world who are experts in protease, viral protease inhibition, or viral helicase inhibition, etc. And many laboratories in the world are going after drugs that are selective for this virus, or effective for this virus, as well as for other coronaviruses and many others. I will just point out that Atlanta has two of the most successful academic inventors of such drugs in Dennis Leota and Ray Shinazi at Emory, both of whom are responsible for more than half a dozen drugs that are on the market for battling HIV, hepatitis B, etc. And so there's lots of local expertise in this area as well as other places. My group, however, decided, as we have done for many years, to work on the immunology. That is, to see if we can get the immune system to make antibodies that will do several things, but one of them is to inhibit that ACE2 binding that you see at the top left. If we can make viral infectivity, make your immune system render the virus unable to infect your cells. Next slide, please. So this project that I'm about to tell you about actually has a more an easier approach in some sense, a faster goal in mind. Our eventual goal, of course, is to create a vaccine. But what is needed, as Loren pointed out, are more and better tests. The RT-PCR based test that you've heard about requires all that entire process. It would be nice if we could detect the virus and detect your immune response to the virus with simple test kits like you would use a pregnancy test, simple paper strip test kit. And to do that, if you want to detect the virus directly, you need to be able to grab onto the virus. If you have a small amount of virus that's present in a throat swab or in a sputum sample, you would want to be able to grab onto it, separate it from everything else in that sample, and then detect it in a simple test kit. And there's many components of that are difficult to engineer, but as you know, paper test kits for a bunch of things are certainly available. And so the key to that is finding the entity that will bind tightly and specifically to the coronavirus and nothing else. So that's what this project is about. Next slide, please. I'm going to skip false negatives. We had a discussion of that. So go ahead and go to the next slide. And so here's the goal. We want to develop these little grabbers, which are the Y-shaped cartoon molecules you see at the lower right. An example of a kind of test kit strip that you might see is on the left. And one of the many ways in which to engineer these kinds of tests is to immobilize things that will grab onto the virus, as you see rendered here very sophisticatedly. and then have something else in the test kit grab onto the viruses that have been grabbed onto and immobilized, and then light them up so that we can say, there's a bunch of virus here that came from my sputum sample, or there isn't a bunch of virus here that came from my sputum sample. And so the Y-shaped molecules in blue and pink there that you see, those are antibodies. In principle, one can have anything bind the virus selectively, but our immune systems are particularly good at developing molecules that selectively bind to pathogens like viruses. And so our project derives from a work that we've been doing for a couple of years with the CDC to develop antibodies to a wide variety of things that they need to detect. The CDC is, among many other things, the world's premier source of diagnostic kits and diagnostic reagents for detecting all sorts of diseases, all sorts of toxins in our environment and in the natural world. And so when you want to validate that you've seen something, you send it to the CDC if you are wondering about a disease, and they will have a test available, or if not, they will make it to detect the pathogen that you're interested in. So these are the best people in the world at this, and we were lucky enough to get connected with them because of our work in immunology and developing a platform to generate high affinity immune responses to a variety of molecules. So next slide, please. And so here's the approach. Here's the way this is done. This is very simple stuff in this game. One creates an immunogen, that is the thing that you want the immune system to see. Sometimes, and of course, many vaccines are like this, they are simply the pathogen itself that is inactivated to make sure that it's safe to handle, but is otherwise the whole ball of wax, the whole bacterium, the whole virus, or whatever. Or it could be components of that pathogen, such as a protein or a part of a protein, formulated in such a way that the immune system can see it. So that inoculation is done of typically a mouse, but it could be other organisms. One follows well-trodden paths of methods, to have the virus, to have the immune system see that pathogen. And then after a certain amount of time, and you check and make sure that the immune response is being mounted, you then isolate the cells called B cells that actually display the antibodies on their surfaces and make and secrete those antibodies. Those B cells are isolated from the spleen of the animal, and in a process called hybridoma fusion, you essentially create a mixed cell type. You fuse them with immortal cells, myeloma cells, by a rather crude process that fuses cells together to impart essentially immortality to the B cell, but retain the ability of the B cell to make and secrete the antibody. So those cells are called hybridomas. That technology is more than 20 years old. Those hybridomas are then, one can store them, one can freeze them, one makes many of them, and then you simply have to find the hybridomas that are producing antibodies, that are producing a lot of antibodies, and that those antibodies bind tightly to the antigen, to the thing that you're trying to bind to. If you do this right, each hybridoma produces only one kind of antibody, only one molecule, but many copies of that one molecule. That would be called a monoclonal antibody. So essentially then one has to test a whole bunch of antibodies or hybridomas to see which ones bind the target the best. And then you can essentially expand, you can make many more copies of that cell, and you then have little factories that make monoclonal antibodies. So that's the pathway. Go ahead and click the button, Sue, and you'll see the partnership then between our lab and the CDC is as follows. So we make the immunogen. Go ahead, next click, please. And we do the immunizations, and we check the immune response and isolate the B cells. And then the CDC takes over. We ship those cells to the CDC across town. They have a marvelous technology for generating many thousands, actually millions of hybridoma cells, and then selecting from those cells thousands of examples of hybridomas that are producing lots of the kind of antibody that we're interested in. And then both Georgia Tech and the CDC, our lab and the CDC lab, then go about the laborious process of screening, that is finding which of those many thousands of antibodies bind the target the best. And that is a wonderful workflow which takes advantage of the laboratories, the strengths of both labs, and we're just having a delightful time. We have been working with the CDC on developing antibodies to some interesting things, such as drugs of abuse or environmental toxins, or sugars and peptides that are characteristic of organisms such as Streptococcus or pertussis. But all of that work stopped 2 months ago. And at the beginning of March, we started to rapidly generate immunogens and apply this workflow to the development of antibodies against the coronavirus. Next slide, please. So there are different kinds of antibodies, but just to tell you that antibodies are large molecules. The Y-shaped versions that you see average about 150,000 Daltons in molecular weight, so lots and lots of atoms there. The little tips of the Y's, of the two tips, that's what's called the combining site. So most antibodies like IgG and IgE and IgD have two binding sites per antibody. And what happens in the immune response, among many other things, is that the sequence, that is the structure of the antibody, is constantly mutated until efficient binding is obtained against the pathogen that is making you sick. That process takes several days to begin, and then several weeks to reach full affinity, if you will, full maturation. It is a process of controlled evolution. And indeed, evolution is the only thing that can beat a rapidly evolving pathogen such as this. So this is why your immune system is constructed the way it is, to evolve rapidly, to produce molecules that bind selectively to whatever is invading it. And so we are looking to generate antibodies that have nanomolar or better affinities to selective affinities to this virus. Next slide, please. So eventually what we want is, among other things, what's called a neutralizing antibody. And here's an example, click again, Sue, from the 2003 SARS virus, and one more, click, thanks. So here's an example of a study, many studies, showing the production of what's called neutralizing antibodies by immunizing mice and isolating those antibodies and finding that either the serum from those mice or the isolated antibody is sufficient when added alone to render the virus non-infectious. And that's called neutralization. So on the top left, you see an example of that experiment. I'm sorry for the small type, but on the vertical axis is the amount of virus that's being produced in this particular assay. And on the horizontal axis, the x-axis, is the concentration of the antibody. And so as an example, these curves are how the antibody binds to the target, and the little filled in dots are the concentrations at which complete neutralization, that is inhibition of the viral ability to replicate, is observed. And so what you see here is that the isolated antibody is about 1,000 or 3,000 times better at neutralizing the virus than just all of the serum, which has antibodies in it, but also lots of other stuff. This is the kind of result you're looking for. You want your serum to be able to deactivate the virus, and then you want the antibody to be really good at deactivating the virus. In the bottom left there, those images, those are actual micrographs of a coronavirus, now the 2003 version. And what you'll see in the bottom of those images, I'm not projecting, so I can't use the pointer, sorry, but the one where you see that scale bar, that 100 nanometer scale bar, what you're seeing is antibodies bound to that virus, and those little black dots are little gold particles that are fixed to the antibody, so you can see where they are. But if you look really carefully around those viruses, you can see these puffy extensions. That's the spike protein that we were talking about. It looks a lot less neatly arrayed than the image that you get from the CDC. But in essence, that's what those spike proteins are. They stick out quite a bit from the virus. In the top right is a cautionary tale from this paper. It shows binding curves. That is, the higher the signal on the y-axis, the more antibody you have bound. Going from left to right means higher and higher concentration, As you go from left to right, those lines should all go up. And so the higher they go up at the lower concentrations, the better the binder they are. However, the black dots or the filled in symbols, dots or circles or whatever, that shows you the concentration at which each of these different antibodies is neutralizing. So what you see is there are a few of those antibodies that have those circles or triangles or diamonds that are way on the left side. And it turns out they don't bind very well to the particle, at least in this experiment, and yet they're neutralizing at low concentration. Whereas others bind tightly, they're way up on the vertical axis, but they don't neutralize until they're 10 or 100-fold higher in concentration. And what this means is that this is complicated. Just finding an antibody is not enough to know that you're going to be able to defeat the virus in the context of a vaccine. And so we're going for tight binding antibodies initially just to find the virus so we can say in 5 minutes that you have it. Making a vaccine is quite a bit more difficult, but this is a good first step. I hope that was clear. Next slide, please. So I'm going to skip through a bunch of this because this is Ashley's amazingly hard work, but the lessons here are only ephemeral in the sense that we're still we're still deeply into this study, so we don't know what the final lessons will be. But at least it'd be nice to give you a flavor, we thought, of how this work is done. So we immunize mice and then we repeatedly immunize them. That's called boosting, and familiar with going back to the doctor's office to get a second immunization, a booster shot. So we do that with animals as well, and normally we like to do it in 2-week intervals, but this is a crisis, and so we are compressing that kind of schedule into an experimental design that most immunologists would recoil at in horror, and we just have no choice. We need to begin to generate these antibodies as quickly as possible. So we're immunizing quickly, and we're doing this kind of antibody selection that you saw earlier on a weekly basis now, quite intensively. Next slide, please. Skip this one as well. Sorry, this is the same kind of information. I took too long in the other part, so I don't have a chance to walk through this. And so here's an example of what we're doing. So we can get, by purchasing them, or our CDC colleagues produce, the full-length protein that is this spike protein. And they can make it by expression in various cell types. What you're seeing here is the structure of the trimer. The form of the molecule we get will vary. Some of it can be trimer like this. Some of it can be just one of those colored domains that's not trimerized. But we can immunize with that simple protein. And so that's, of course, the first thing that we started to do. Simple experiment, buy the protein, stick it in mice. Of course, we do this in a way that is designed to stimulate the immune system, but that's essentially it. It's a total shot in the dark, as it were. There's no real hypothesis-driven science here. It's, we got the protein, let's see if we can raise antibodies. Now, there are, I could imagine hundreds of laboratories doing this worldwide, doing this exact experiment. The reason why it's worth doing it here is that this is a random exercise. The immune system is going to see what it sees, and each animal will be different. Each selection process will be different. And the more people who do this, the more chances there are of finding the right antibodies that can then be produced at scale to provide test kits and maybe even therapeutics after a long process. So we don't have to be unique here. We just have to contribute to the worldwide effort, and hopefully somebody will stumble on one or many antibodies that fit the bill. So we're certainly doing this. Next slide, please. Another way that we do this, which is unique, or at least we're doing it, not everybody can do this, is we display these proteins on viruses themselves that are not the coronavirus, but the viruses that we use similarly wake the immune system up to saying, hey, there's something that looks here like a pathogen, I need to pay attention. So we can take this protein from the coronavirus and essentially fuse it to our virus. and immunize with that. And so we're doing that. Next slide, please. And then these are the kinds of results that one gets, that you're beginning to get. And I'll try to give you a flavor for this experiment. You immunize with something, and then you collect a little bit of blood, a little bit of serum from the animal, and you have to ask, is in that serum, any of the antibodies in that serum, do they bind to my target? And so you need to put some form of the target on a plate so that you can pass the serum over that plate, wash away anything that doesn't bind, and then come in and ask, are there any antibodies from the animal that remain bound to that plate? So what we put on the plate matters a lot. And so pretty much all of the data I will show you is, no matter what we're immunizing with, on the plate we put this spike protein. And so what we're looking for are antibodies that we produce that bind to this spike protein. And that spike protein can be in different forms. We're doing this rapidly, so we're plating now, we're probing, as we say, with the only form of the protein that we could get initially, which is just one of the domains of this spike protein. As we and others produce more refined forms of the probe reagent of this target, we will reproduce this measurement on more and more relevant forms of what it is we're going after. Ideally, of course, we would be putting down some inactivated form of the virus itself to make sure that we're generating an immune response to that whole particle, or to some part of that particle. So this is the kind of data you get. Concentration goes down from left to right, and binding is on the vertical axis. So you see these S-shaped curves, which shows you that you have tight binding, and the more you dilute the serum, the binding goes down. Exactly what you expect to see. The farther the curve extends to the right, the better off you are, because that means the more antibodies you have, or the higher affinity antibodies you have. And so I am happy to report that at least the initial rounds of experiments, some of that stuff we're looking pretty good. And so the CDC lab now has made the hybridomas, and we're getting ready to do our first several rounds of screening thousands of hybridomas to see if we can pull out the best antibodies from just this first round of immunizations. Next slide, please. Here's an example. No, I will actually skip this slide. It's just more of this kind of data, just to show that we make millions. This is what this slide shows. Millions of these hybridomas can make. And this is not unique to them, but they're very, very good at this. Next slide, please. So another approach that we take, we're labeled here AIM2, is to divide up the protein by looking at its structure and ask which parts of the protein, which amino acid sequences, are unique to this coronavirus versus any other virus, and which might be well recognized by the immune system. There's a number of criteria here. Physical exposure, the immune system will see initially only the things that are on the outside of the virus. And so physical exposure is one, and then the peptide sequence it will depend. The peptide sequence will determine how well the immune system can process the protein or that peptide sequence and evolve an immune response to it. I'm not going to go into the details there because I've gone on far too long already. Suffice it to say that we and many others have analyzed the sequences of these proteins in great detail and come up with a selection of peptides that we think are uniquely good candidates for immunization. So the next slide, I think, will show you some of this selection. Yeah, so here are examples of peptide sequences that we've picked out, and the names of the people doing these analyses are here. All of whom are wonderful folks in my lab. And as an example, you see the sequence of 6 or 7 of those peptides pulled out on the right. And the style of immunization is then to create particles that display that peptide sequence in multiple copies and give it to the immune system in a way that says, hey, look at me, you should evolve an immune response to this sequence. So that's the kind of thing that we do routinely using our particular virus display platform. So next slide, please. That work is now in full swing. And the color coding in the bottom left, you can see, is where those peptides are on this trimer that is displayed in multiple copies on the viral surface. So you see we've picked out peptides that are all over the place, which is what we want to do on the outer surface of the spike protein. Next slide, please. This is characterizing the virus particles that we produce, not coronavirus, but the display virus particles. I'll skip this, but there's a lot of work here on making these particles and characterizing them and producing them in very pure form. We do that very well. Langzhen is a master. And next slide. And then the immunology is done in exactly the same way. In this case, we're immunizing with the particle that displays the peptide, and then we can come in and boost with the full protein from coronavirus and ask the immune system to essentially focus on the peptide that is in common between those two forms. We deliver the peptide on one virus, we come in with a protein that has the same peptide, but in the context of the viral target. And we're hoping that the immune system says, I started to look at you and now I see you again, and so I'm going to focus on you, the you being the peptide that we're hoping is presented in the right way. You'll notice a lot of the language here is we hope, we're trying and all this. This is because, again, we have little control over exactly what the immune system does. We think we can try to direct it in one way or another, but we have to run a lot of experiments to give the immune system a lot of tries at this. Next slide. Almost done. And so here's an example of when we immunize with a peptide and boost with a full protein, we're getting some response. This is actually tremendously encouraging. Two of these peptides, number 6 and number 16, seem to begin to elicit an immune response that is then reconfirmed by the full protein. Other peptides that you see on the right there give us no response, which is actually good. We know that we're getting some specificity when we see it. Next slide, please. And then we turn the experiment around, and when we immunize with the full protein and then we came in with the peptide, we see one of those same peptides lights up. That's this plot on the left. We see this blue curve going up. That means that when we immunize with the full-length protein and we ask it, does that immune response see any of these peptides that we have displayed on a different particle? One of those peptides is recognized by the antibodies produced to the full protein. So this represents our first really cool lead as to a focused peptide antigen that we might have a chance at amplifying into a coronavirus-specific response. Next slide, please. I'm going to skip this. This is another target we're going after. This is in progress. It's the same kind of approach, but here we're targeting specifically the receptor binding domain. But skip this one because I have no results to show. But I did want to show this. So somebody asked after Loren's talk about mutation and about the viability of the primers that are being used. The mutational history of this virus is being tracked in pretty much real time. This is a wonderful website that you can go to, and it doesn't take too much to figure out how to read this. But here are two of the plots from that website that I just downloaded today. What you're seeing here is if you start at the left, there's a single purple horizontal fragment of a line. That represents the first coronavirus, first novel coronavirus that was sequenced. And then it begins to diverge. And as you go from left to right, you see these branches. At each branch point means a mutation has been detected in the viral genome. So what this is a historical record, because the x-axis is the date, of the mutations from the original virus that was characterized. This is, of course, not the original virus, but the first one that was sequenced. And so you see there are now 3,000, almost 4,000 mutant viruses, the sequences of which have been characterized. Don't be alarmed. This happens with every virus, and this one is no worse or better than the rest in terms of its mutational frequency. It's actually kind of slow. But go to the next slide, and you'll see another reason for not tremendous concern. And that is that the number of mutations is actually so far not so high. So you'll see it's hard to read on on the x-axis, but we're up to only about 20 or so mutations for the most highly mutated virus. And that's over a period of 3 months or so. Now, if one of those mutations is in a key component of the virus, for example, like the place where our antibody might bind, well, then this virus is going to be easy to defeat this particular antibody that we might be developing. So the site of mutation matters a lot. This is room for concern, as it always is in viral immunology. This is what viruses do. And so the take-home message here is this virus is mutating. We don't know how bad those mutations are with respect to therapeutic or immunological intervention. And we're going to learn that over the next months and years. So I'd be happy to take questions on this. This is why we need a new flu vaccine every year, for example. But a lot of the questions I think you're going to ask, or you could be asked, we just don't know the answers to. So I think that's it. Next slide, please. I think is the last one, to just make sure I repeatedly acknowledge Ashley Chapman, amazing immunologist and biochemist, and she's just doing a ton of work. Lang Jun, a protein designer and producer par excellence. Jeff Noble, marvelous immunology as well. Robert Incapier will soon be pressed into service to work his bioconjugation magic to make various immunogens using synthetic peptides. And then Jason and the rest of the immunodiagnostic development team at the CDC are really the world's best. They're phenomenal. So thanks, and I'll try to answer some questions.
There's a couple of questions, one from Richard Bedell or Bedell and Luna One. They're asking, their questions are related to what about antibodies that are produced by people? How does this fit into this workflow that you've described in mice?
Right. So great question. So the antibodies that we develop in mice obviously can be used on a paper strip, right? Because they're not going into people. You can, however, develop an antibody in a mouse and then do what's called humanizing it. If you take a mouse antibody and inject it into a human body, you'll get an immune response and an allergic response, and it's not good, right? Because our immune system knows that molecule came not from you, but from another organism. However, one can humanize mouse antibodies or any antibodies by knowing the sequence of the antibody and then changing the portions of the antibody that don't bind to the target, because those are not what's detected as being foreign, but change the part of the antibody that swap it out for basically a human antibody component so that you can take those antibodies and give them to people. And so our molecular evolution core facility, headed by the marvelous Anton Brixen, has worked out a method to sequence these antibodies rapidly, given just a few cells that produce them. And so we have the pipeline in place to sequence, and then the CDC is very good at humanizing the antibodies that they get. So it is possible, as we all know, to use antibodies as a therapy, not just to immunize, to engender an immune response, but to give antibodies as a medicine. And this is, of course, a lot of drugs are antibodies now. You have to inject them, you can't eat them, but that is a therapeutic possibility.
Jennifer Hill Lum asks, there's a vast amount of work being done and data generated. How is all this information going to be shared with the global scientific community as in real time as everybody's working on this?
So lots and lots of people are posting papers to various archives, right? ArchiveX and others. So you'll see a huge amount of information being published, freely accessible, not peer reviewed, but that's okay. And so we plan to do this, and pretty much everybody else is doing this kind of thing. So all the sequencing information is being dumped into publicly accessible databases. It's really a wonderful global response, scientific community.
Professor Russo asks, 5 repeat units at once in the RBM seems like a lot, a sudden change. Is that why no one has immunity? Will all the work that you do hold up to future mutations better or worse than the usual flu viruses?
Yeah, great question. And the answer is we just don't know. So this virus is novel, of course, because it hasn't made its way to the human population yet. And the mutations in the RBD domain are not necessarily why it's novel, but it is an important component of why it's so infectious. But we don't know the mutational frequency in the residues that matter. And of course, a lot of the game here is to find the parts of the virus that don't mutate rapidly, the thinking being those are the components of the sequence that the virus needs in order to be infective, right, in order to reproduce. So you find those relatively invariant sequences, and then you see if you can create an immunogen to go after those. That has been the problem with HIV vaccines, among other things, for decades now. And HIV is also a problem because it's covered with a dense coat of carbohydrate, as coronavirus is. And I didn't talk about that, but that's, of course, potentially an issue. So finding the invariant portions and making them immunogenic in such a way that the immune system can see them and that antibodies can get at them, that's the trick. And so the answer is, sorry, Paul, we don't know.
There's a question from Mofu Dominic, who's asking, do you know what type of method is being offered for the rapid testing that's currently taking place on our campus?
So that's a variant of what Loren talked about. So that's an RT-PCR-like test. So you take a swab, that material is then digested so as to be able to extract the viral RNA that may be in there, and then that RNA is amplified with a machine that they have. That's actually many copies of that machine in the parking deck here. It's the test that was developed by Amgen. And I will brag a little bit, as I have to others, that one of my former students at Scripps was the co-leader of that effort to develop that assay at Amgen. But it's using an Amgen instrument and a proprietary procedure. But it is, in essence, an RT-PCR assay.
Mm-hmm. And I think there's one last question. Do you still have a couple more minutes, MG? Are we good on time?
Yep.
The question is from Bo-Chung Wu. How are we sure that the mouse can generate monoclonal antibodies or not?
Well, the mouse will generate antibodies. We make them monoclonal by finding the B cells that producing them, making hybridomas, and then most of those hybridomas, the vast majority, produce a monoclonal antibody because they produce only one antibody. So the mouse will, the real question is, will any of the antibodies, or will a sufficient fraction of the antibodies produced by the mouse have the properties we need? Will they bind to the target with high affinity and selectivity? Will they be neutralizing? Will they induce a type of response after the immune system finds them so as to kill the pathogen? Those questions, it really is, by and large, pretty much pure chance. So we've just got to try a whole bunch and analyze a whole bunch of hybridomas. This is why this partnership with the CDC is so exciting, because they have technology that allows us to do that in a very large way. And so, but it's a crapshoot.
Well, thank you so much. I'd like to thank the speakers, Professor Finn and Professor Williams, for taking the time to tell us about all the things that are happening in our department. It's really amazing to know that we can contribute to this effort in such a meaningful way. So thank you. Thanks to Sue Winters, who helped facilitate this through her outstanding technical abilities. So thank you, Sue. And please sign up for giving a talk for our virtual seminar series. There's a link going around that I'll continue to send out, but hopefully we can meet again sometime in the next couple of weeks and continue this tradition. So thank you all to the speakers and to all the attendees. Night, everybody.
Thanks, Amit.